Hello, my name is Lexis Caputo. Today's date is November 15th, 2018, and today I will be performing bacterial morphology and staining techniques for Bio 225, Week 3 assignments. This portion of the lab is to perform staining techniques, and the first sample is a sample direct from the mouth, and this is the first one. So right now I'm just letting the smear cool after passing it through the flame for 5 seconds to allow the bacteria to adhere to the slide. And then after 10 minutes, I'm going to apply this crystal violet stain, in which is going to sit for 60 seconds. And then we're going to rinse it and then apply the next set of stains. This is the second portion of the staining technique, in which we put a drop of the Congo red just under the label, which is the second dye, and it's a negative sample or a negative stain. And so we put the drop of the Congo red just under the label, and then you took a sample again from the gums and teeth area, and then we put the sample in the stain and kind of agitated so that the cells from the swab transferred to the slide, and then you use another slide to remove the excess liquid, and now we're just waiting for it to dry in order to observe under the microscope. The third exercise of this lab was to practice gram staining, in which all 3 of the bacteria first received a crystal violet, in which sat for 60 seconds and then was rinsed with tap water, and then received iodine, and then was rinsed after 60 seconds, and then received decolorizer, which sat on the samples for 30 seconds, and then after that was rinsed, the safranin was left on each culture for 30 seconds and then rinsed, in which this process is to determine whether a culture is gram positive or negative, in which gram positive cultures have a thicker wall, so they will keep the crystal violet dye, in which the E. coli culture seemed to do, and then the Staphylococcus epidermidis had the red tint, and then the Saccharomyces cerevisiae actually kind of remained colorless throughout the portion. So just by going through the experiment, it seems as though the E. coli is gram positive, where the Staphylococcus epidermidis would be negative, and then based on the experiment, the Saccharomyces cerevisiae seemed gram variable, as no color remained after the staining.